Hello, and welcome back to another exciting episode of What is a Biology? I'm your host, Mr. Johnny Hopkins, and today we're going to be talking about DNA and RNA, just their basic, simple structure. So, what is a DNA? What is an RNA? A DNA is a deoxyribonucleic acid, and an RNA is a ribonucleic acid, or, if you will, an oxyribonucleic acid, because it's not deoxy. So, there's a lot of things in DNA, and it's made up of these constituent parts called nucleotides. And I've drawn out here the molecular structure of a nucleotide. This polymer is a little bit crazy to look at instantly. So it's like a protein. It's made up of its constituent parts, and it's got a side chain that is that varies. But instead of the 20 different possible side chains for proteins that are strung along, you only have 4 for DNA as well as RNA. And so a single unit, a monomer of this DNA is this guy, something called a nucleotide. A nucleotide includes 3 things: a phosphate, a sugar, and a base. And then also you can get a little bit more specific and includes something called a nucleoside, which is just this guy. Nucleoside, nucleotide. So the nucleoside is just this pentose sugar and the base itself. So what happens here? So we have this guy go from phosphate to sugar to phosphate to sugar to phosphate to sugar to phosphate to sugar to phosphate to sugar, and so on and so forth for a long time. In the human genome, there are about 3 billion bases in the human genome. So if you stretch this all out end to end, you get about 2 meters, which that's kind of long for just a single cell. So just pluck out a single cell and you got a really long polymer sequence. So there's a lot of things that are going to come into play with having this long, huge sequence later on. But rather, we're going to just focus on real quick onto just what this DNA looks like. And so you have this chain right here, and then you have another chain usually right by it with the DS double-stranded DNA. So it's the end of the DNA that ends in a phosphate is called the 5' end. And the part that ends in the sugar, or with the usually oxygen right here, is the 3' end. It's just a notation. It's nothing particularly interesting or really noteworthy besides just understanding where things are. So you have the 5' end is the phosphate end, and the sugar end is the 3' end. And how does DNA make this double-stranded helix that we've always been hearing about? And the way it does is by 2 things: hydrogen bonding and van der Waals forces. We'll get a little more into that a little bit. But so, as we kind of know, there's this Watson-Crick base pairing scheme that has continually happened, is that you have a base on one side of the helix, on one strand, hydrogen bond with the other side. So this amine group hydrogen bonds with this oxygen, because amines are usually positive, this guy is usually negative. And then over here we have this nitrogen bond with this nitrogen right here, because this hydrogen is slightly negative, this nitrogen is slightly positive. And then over here we have a similar thing with the amine and oxygen, same thing over here. So I have 3 base pairs in this guy, which is a cysteine-guanine interaction and an adenosine-thymine interaction right here. So that's the basic structure. You have monomers of these nucleotides, which is composed of a nucleoside, and the bases hydrogen bond with each other. And that allows these things to stay nice, tightly connected, and make sure that our body functions, and we can go and pick up markers and scream in the middle of a camera on YouTube with nobody listening. So how is this DNA arranged in a cell? And that question is a bit strange because it's not the same way in every cell. Typically the way to look at it is eukaryotes versus prokaryotes. So eukaryotes are things like humans, dolphins, millipedes, these animal structures. And then you have prokaryotes, which are like bacteria. Just you know, the things that you don't want to get in your system because they'll infect you and make you sick. So eukaryotes, you have these guys are lined up into a single strand of our in chromatids, and it's just kind of a long linear polymer that has some massive tertiary structure. And then you have prokaryotes, which is just a giant circle essentially. This DNA is just a giant circle. Then you usually have some smaller circles called plasmids that also encode different things. And what DNA encodes is something called a gene. And a gene is just a hereditary unit controlling identifiable traits. Welcome back to Socrates and the art of semantics. This tells us practically nothing. Essentially, a gene is just something that gets transcribed into RNA. So, what is RNA? RNA is just usually a single-stranded thing that looks a lot like just one of these DNA strands, except it's added on a hydroxyl group onto this part of the nucleotide / nucleoside. It's got the same 5' end with the phosphate with the nucleotide. So that's the nucleotide, and the nucleoside is just the same pentose sugar and base. So, bam, bam, nice. So it's a lot like this, but you've got that oxygen right there. And that oxygen makes it a lot more susceptible to attack and degradation. That makes it chemically a lot more of a temporary solution, which is very good for the cell because it allows for a lot of moderation of RNA in the cell. So, real quick, we're gonna one of the basic things about RNA and DNA is if you look at this, you might think something quite simple. This phosphate, that's a negative. That's a negative. That's a negative. That's a negative. You have all these negative charges stacked up right next to each other. They're wanting to butt up against each other and fight each other. They are ready to go. UFC, MMA, fight their neighbors. It's like the Civil War. So you have brother against brother. And so how do we take into account, that's not supposed to be negative, my apologies. How do we take into account how these negative coulombic charges interact with each other? And typically, this is done by something called magnesium. It's an ion that usually carries a 2+ charge, and it just sits right next to this phosphate backbone, and because it's got a positive charge, it nullifies the coulombic interactions and makes it stable. So it's why we have DNA in the cell, tell the central dogma, and make us think about applesauce all the time. And so, how do we get from DNA to RNA to proteins to make us think about applesauce? And the way we do this is that there are typically the most common 3 types of RNA discussed. There are a lot more different types of RNA, but these are the 3 ones that are most commonly discussed. mRNA, tRNA, and rRNA. mRNA is a transcript that's made from DNA that gets made into a protein. Then you have rRNA, which is ribosomal RNA, which its job is to make the ribosome, which takes mRNA and turns it into the protein. Then you have tRNA, which actually interacts with the rRNA and mRNA in order to incorporate a peptide chain and make a wiggly boy protein that eventually becomes an actual mature protein. So one thing that's interesting about RNA is because it's single-stranded typically in nature, whereas DNA is double-stranded, it actually likes to form bonds with itself. It likes to give itself some self-love. Live, laugh, love is the motto of mRNA. So what happens is usually the RNA will actually hydrogen bond with itself. So you take a wiggly boy RNA, and then it will actually form hydrogen bonds with itself, and then form a secondary structure that looks kind of like this, where you have hairpins sticking out of it. And so one actually interesting thing about rRNA is that it's a ribozyme. And a ribozyme is just RNA that catalyzes some sort of chemical reaction. We'll discuss more about that when we talk about the ribosome. So, now, as I previously discussed, there are different bases on these DNA and RNA. And there are in total 5 canonical bases used. You have the 2 purines and 3 pyrimidines. Purines have 2 rings, pyrimidines have 3 rings. Sorry, have 1 ring, my apologies. And we have guanine, which has the chemical structure of this, which base pairs with cytosine. And this is in DNA and RNA. You have adenine, which base pairs with thymine in DNA, but adenine and uracil base pair in RNA. It's just the difference between RNA and DNA. So if you just take notice real quick, the only difference really is right here, where you have a methyl group being added on in thymine, whereas you just have hydrogen right there. Not a huge difference because it doesn't actually change the binding interface of the hydrogen bonds with adenine. So that's the bases that can be put onto mRNA and DNA. Then finally, how the primary interaction that actually allows for DNA to hold together is not hydrogen bonding, which is a common misconception because of the importance of these Watson-Crick canonical base pairs, but rather it's base stacking. It's van der Waals forces, which if you just want to go real physics on it, it's just electromagnetic forces. So like everything is electromagnetic forces, but if you need to be more specific, van der Waals forces, specifically base stacking, such that it's like a zipper. You have a base pair and a base pair, such that they line right on top of each other, such that the thing that holds it together is not actually the interaction here, because that can kind of just fall apart, if it will, but everything on top of it and everything below it keeps it squished together so it doesn't want to move, like a zipper. Because the things that hold it together are not the interaction with the side, but everything on top and everything on bottom. It's a zipper. And that causes base stacking. So, if we look at it, you've probably seen pictures like this B form right here. And there's good reasons for it. So, what you have is 3 different forms that DNA and RNA can take in a double-stranded nature. You have A form, B form, and Z form. We'll first just cover Z form because Z form is the weird form and it's not really seen in vivo. It's usually just this in vitro mixture of strange stuff happening where like this backbone is just kind of disgusting and mangled, and it typically just happens in double-stranded DNA with an alternating pyrimidine and purine structure, typically with the guanine cytosine structure, such that it gets this weird backbone that you're not going to really talk about ever unless you're really, really into nucleic acids. Then you have A form and B form, and so we'll go and talk about B form because this is the canonical, typical DNA you're going to be talking about. Double-stranded DNA, it's always in B form. So this is DNA-DNA interactions. And there's some interesting stuff. And what you have is something called a major groove. Major groove. And a minor groove. And so it has this major groove that goes and wraps around. So this is part of the major groove. And if you took it around in a 3D structure, you would be able to actually kind of visualize and see that this guy is the same part as this guy, is just kind of following in a little spiral, as well as the minor groove is itself its own little spiral. Oop, that was very bad of me. That's the major groove as well, but here is the minor groove. So the minor groove, it topologically is the exact same here as here as here. So if you just look, it's like a ridge. And so going from major groove to major groove, like just like this little segment right here, you have it's 10 base pairs, and so which translates to 34 angstroms, which translates to 3.4 nanometers in length, because the distance between a single base pair stacking on top of each other, so this distance from here to here is 3.4 angstroms, such that they stack on top of each other and are 3.4 angstroms away. So that gives enough for van der Waals forces to keep them in line. And so B form, it's a long, slender structure, and the bases are close to perpendicular with the helical axis. They're slightly tilted, but nothing too major. And then you have A form. And A form is typically formed if RNA, double-stranded, as well as an RNA-DNA hybrid. And this is a wide structure that is much wider than the B form, also still has a major and minor groove, but most notably, the bases are actually tilted against each other with the tilted against the helical axis. So they tilt and tilt, especially when compared to B form. Typically, you will mostly just see DNA nucleic acids in B form, because you're typically talking about DNA-DNA interactions inside the cell. So it's not a bad idea to just have the idea of a B form DNA at all times, and then when someone clarifies, you can think of A form, or in the very rare case, Z form. So, all in all, DNA is made up of a string of nucleotides going from the 5' phosphate end to the 3' sugar end. And it's made up of 4 different bases: guanine, cytosine, adenine, and thymine, all of which base pair with each other in hydrogen bonds, as shown. And these hydrogen bonds help form the correct canonical base pairs, as well as help form into a base stacking where they allow for a stable structure. RNA is the same thing pretty much as DNA, except it's single-stranded in nature and has an extra hydroxyl group, which makes it a lot more chemically active and likely to be degraded in the cell. And because it's single-stranded, it likes to fold in on itself and form these weird secondary structures with multiple hairpins inside it. Another difference between RNA and DNA is that you have adenine base pair with uracil instead of thymine. And then finally, RNA-RNA interactions make into an A form, which is a slightly more tilted base pair interactions with the DNA helix. Thank you very much. That has been What is Biology, talking about DNA and RNA structure. Next time we'll go more and talk about the transition from DNA to RNA, known as transcription. I'm Mr. John Hopkins, and this has been What is Biology. Thank you.